If you look around at the sheer, uh, just the mind-boggling complexity of life, I'm talking about everything from those golden-furred, quinling, snub-nosed monkeys in China
Hmm.
to, the yeast growing in a lab,
Hmm.
you're seeing this almost infinite diversity of structures.
Right.
But how do you get all of that complexity from just a handful of common elements?
That is the foundational question, really. And the answer, the master architect behind all of it is a single atom. It's carbon.
So that's our deep dive today. We're getting into organic chemistry, the study of compounds that contain carbon.
Exactly.
And our mission here is to really break down the three core ideas that explain why life is built on carbon. First, carbon's unique versatility.
Mm-hmm.
Second, how that versatility leads to all this complex molecular architecture.
Mm.
And third, how these tiny chemical attachments, the functional groups,
Mm.
are what give a molecule its actual job in a cell.
We're basically looking for the chemical fingerprint of life's diversity.
Perfect. So where do we start?
We have to start at the very beginning with the elements themselves. Living matter is almost entirely made of carbon, hydrogen, oxygen, nitrogen, sulfur, and phosphorus.
CHON, S, and P.
Right. And while the percentages of those elements are surprisingly uniform across all life, which is, a huge clue about our common ancestry, it's carbon's genius for bonding that makes everything else possible.
It's genius for bonding. I like that. So structurally, carbon has 4 valence electrons.
Right.
It needs 4 more to complete its outer shell, so it forms 4 covalent bonds.
Hmm.
Most other key elements are limited to 1, 2, or maybe 3 done.
And that's the key. Carbon can act as an intersection point, a hub. It lets molecules branch out in up to 4 different directions. It's the ultimate biological scaffold.
So you have this amazing scaffold builder. The next question is, how did it start? Can that kind of complexity just happen on its own?
A huge question. And in 1953, Stanley Miller and Harold Urey designed a landmark experiment to try and answer exactly that.
Okay, let's try to visualize their setup because it's become so iconic in biology. Miller created this closed system to mimic the conditions on early Earth.
That's right. He had a flask of boiling water at the bottom, which was like the primeval sea. That created water vapor.
And that vapor traveled up into a larger flask, the atmosphere flask.
And inside that atmosphere, he put the gases they thought were present back then, so hydrogen, methane, ammonia, and then came the energy source.
The lightning.
He used electrodes to create electrical sparks inside the gas mixture. Then the whole system was cooled by a condenser, and whatever formed would drip back down into the sea.
So for a week, he was basically just boiling and zapping these simple non-living compounds. And what he found in that flask at the end of the week was, oh, it was revolutionary.
It really was. Abiotic synthesis, the creation of organic molecules from non-living stuff, it actually happened. Spontaneously.
What did he find exactly?
He found simple compounds like formaldehyde, sure, but he also found much more complex molecules, including hydrocarbons and even amino acids, the building blocks of proteins.
Which strongly suggested that life's raw materials could have just formed on their own through natural planetary processes.
Exactly. Now, of course, our understanding of the early Earth's atmosphere has changed a bit since the 50s.
Right, I was going to ask about that. Some scientists now think the atmosphere was different, maybe more like what you'd find near volcanic vents. Does that kind of invalidate Miller's work?
Not at all, and that's the fascinating part. Later on, researchers reanalyzed some of Miller's other experiments from that era, including one from 1958 where he added hydrogen sulfide to simulate volcanic gases.
Why?
And it worked too. It produced organic molecules, including the amino acid methionine. So the takeaway is that carbon is just so good at this. Organic synthesis can happen under a whole range of conditions.
Which feels, I don't know, almost universal, like the 2018 NASA finding of carbon compounds on Mars.
Precisely. If you have carbon, water, and an energy source, you've got the potential for the raw materials of life.
Okay, so carbon is the foundation. How does it actually use that bonding power to create the incredible variety of shapes we see?
It all comes down to geometry, molecular architecture.
Let's picture the shape. When carbon forms 4 single bonds, like in methane, CHO, the structure isn't flat, it's not a square.
No, it's a tetrahedron. The bonds point away from the central carbon towards the corners of an imaginary pyramid. This gives the molecule a real 3-dimensional depth.
But that all changes if you introduce a double bond.
It changes completely. A double bond, like an ethene CH arrow, locks the atoms in place. It forces all the attached atoms into the same single flat plane.
So you go from a flexible 3D shape to rigid 2D shape.
And that difference, 3D versus 2D, flexible versus rigid, is everything. Because in biology, a molecule's shape determines its function.
So the main source of all this molecular variety comes from the carbon skeleton itself, the backbone of the molecule. We think of them as simple chains, but they can vary in 4 key ways.
Right. First is just length. Ethane versus propane. Easy enough.
Second is branching. You can have a straight chain, like butane, or a branched one, like 2-methylpropane. Same atoms, different layout.
Third is the position of a double bond. You can have 1 butene or 2 butene. Again, same atoms, but the double bond is in a different spot, which changes the shape.
And fourth, they can form rings, like cyclohexene.
And all those variations create this huge library of starting shapes.
The best example of the skeletal foundation has to be the hydrocarbons.
Absolutely. Hydrocarbons are just carbon and hydrogen. That's it. And they're really important because they introduce the idea of nonpolarity.
Meaning they're hydrophobic, they don't mix with water.
Exactly. Think about petroleum, which is mostly hydrocarbons. It just sits on top of water.
And this is hugely important in our own bodies, right?
Right.
Our fat molecules, which we use for energy storage, have these long hydrocarbon tails.
That's why they work so well as fuel. That non-polar nature makes them pack together really tightly away from the water in our cells, so they're a very concentrated energy source.
Okay, this brings us to a concept that, for me, really shows how incredibly sensitive biology is to shape, and that's isomers.
Isomers. So these are molecules that have the exact same molecular formula, same number of atoms of each element, but they have different structures. And because their structure is different, their properties are different.
And the potential for variation here is just, it's mind-blowing.
Yeah.
The book is an example of a hydrocarbon with just 20 carbons, COJ.
Uh-huh.
It says there are 366,319 possible structural isomers.
Over 300,000. It's an astronomical number.
Mm-hmm.
Nature has this enormous toolbox of potential shapes to work with, and that's just for one type of isomer.
Right, structural isomers are just the first type. They just differ in how the atoms are covalently bonded, the branching pattern.
But then you have cis-trans isomers.
Also called geometric isomers.
Right. And these only happen when you have a double bond, because that double bond, as we said, locks rotation.
So if you have 2 of the same group, let's just call them X, attached to the carbons in the double bond, it matters where they are.
It matters a lot. If both X groups are on the same side of the double bond, that's the cis isomer. If they're on opposite sides, that's the trans isomer. And that tiny change can drastically alter the molecule's overall shape.
And we see the effects of this all the time. The difference between a healthy, natural, unsaturated fat and an unhealthy synthetic trans fat is often just this. A cis bond got flipped to a trans bond.
Our vision, too. The whole process of sight is triggered when light hits a molecule in our eye called retinal and causes it to flip from a cis to a transform. That shape change starts a nerve impulse.
Incredible. Okay, but the final type of isomer is where the specificity gets, well, almost spooky. Enantiomers.
Enantiomers. These are isomers that are perfect, non-superimposable mirror images of each other. The defining feature is an asymmetric carbon, a carbon that's attached to 4 different groups.
The classic analogy is your hands.
Exactly. Your left hand and your right hand are mirror images. They have all the same parts, but you can't fit your left hand into a right-handed glove. They're different.
And in biology, the binding sites on cells are like those gloves. They're incredibly specific. They'll often only recognize one version of the molecule, the left-handed or the right-handed one.
And this is absolutely critical in medicine, in pharmacology. Take ibuprofen. It has 2 enantiomers, an S form and an R form.
Okay.
The S form is about 100 times more effective at relieving pain. The R form is mostly useless.
Wow. Or the asthma drug albuterol.
Same story. The R enantiomer is what opens up the airways. The S enantiomer can actually work against it. It just shows that life is exquisitely tuned to molecular architecture. The fit has to be exact.
So once you have the skeleton and the 3D shape, what's the final piece of the puzzle that gives the molecule its function?
Yeah, that's where you add the specialized attachments that dictate its chemical personality. These are the chemical groups attached to the skeleton.
And the best example has to be the sex hormones, testosterone, and estradiol.
It's the perfect example. They have basically the same carbon skeleton, 4 fused rings. They're almost identical.
But they differ only by a few of these attached chemical groups. And those tiny, tiny differences are responsible for, the profound differences between male and female sexual characteristics.
When those groups are the parts that actually participate in chemical reactions, we call them functional groups. And most of them are hydrophilic.
Meaning they love water, They increase the molecule's solubility.
Which is essential since a cell is basically a bag of water.
Okay, so rather than just list them, let's group them by what they do. First up are the groups that are polar and help with solubility. That's the hydroxyl and carbonyl groups.
That's a good way to do it.
Yeah.
The hydroxyl group, O-azarH, is what you find in alcohols. It's polar, and it can form hydrogen bonds with water. Very important.
And the carbonyl group, you see that a lot in sugars.
Yep. Next up, you've got the groups that carry an electrical charge. These are fundamental for proteins. We're talking about the carboxyl group and the amino group.
The carboxyl group -COH.
Right, which acts as an acid. It can donate a hydrogen ion, an H+A, which leaves it with a negative charge. At the pH inside a cell, it's almost always in that negatively charged form,
And its counterpart is the amino group -NH.
which acts as a base. It picks up an H+ from the solution, so at cellular pH, it has a positive charge. And you cannot build a protein without these two. Every single amino acid has to have both a carboxyl group and an amino group. They're non-negotiable for life.
Okay, then we have a couple of groups with more specialized roles.
Like the sulfhydryl group, -SH. Two of these can react to form a strong covalent bond, a crosslink, that helps stabilize the folded structure of proteins. It's like a molecular staple.
And then there's the odd one out, the methyl group -H.
Right. It's the only one that's not reactive, but it's a hugely important tag. The cell attaches methyl groups to DNA to affect which genes get turned on or off. It's a key regulator.
Finally, there's one we have to spend a bit more time on. The phosphate group.
Oh, yeah. The phosphate group, Ajagiro, it carries a big negative charge, but its real claim to fame is its central role in energy transfer.
And this is where we get to the energy currency of the cell. ATP, adenosine triphosphate.
The absolute star of the show. ATP is a molecule made of adenosine attached to a string of 3 phosphate groups.
And those 3 phosphates are all negatively charged, so they're kind of repelling each other, right? They're unstable.
They're very unstable. It's like a compressed spring. The energy isn't really in the bonds, but the whole setup stores the potential to react with water.
So when a cell needs to do work, contract a muscle, anything, it uses ATP. How does that reaction actually release the energy?
The ATP molecule reacts with water. It's a hydrolysis reaction. The bond to the very last phosphate group breaks.
So ATP becomes ADP adenosine diphosphate plus 1 free phosphate ion.
And that reaction releases a burst of energy that the cell can immediately harness to do work. It's this beautiful universal mechanism for linking the energy from food to the energy needed for life.
So as we look back on all this, it's really a story of complexity being built from a few simple, elegant rules.
It is. Carbon's ability to form 4 bonds gives you infinite skeletons.
Then isomers prove definitively that the specific 3D structure is what determines the function.
And finally, the functional groups are the chemical tools, the polarity, the charge, the energy potential that let those structures actually do work inside the watery world of a cell.
We said at the beginning that life is unbelievably sensitive to this molecular architecture. For a final thought, let's go back to that idea of the mirror image.
Okay, let's think about the enantiomers L-DOPA and D-DOPA.
Right. L-DOPA is a drug used to treat Parkinson's disease. It's very effective.
It is. It can relieve rigid paralysis.
Right.
But its mirror image, D-DOPA, which has the exact same atom, same mass, everything, is completely and utterly inert in the human body.
It does nothing.
Absolutely nothing at all. And that, I think, is the ultimate proof. Life doesn't just prefer a specific structure, it demands it. The difference between a life-changing treatment and a useless powder is nothing more than the arrangement of 4 groups around a single carbon atom.
It really shows you that all the biological complexity we see is governed at its core by the fundamental rules of chemistry and physics.